anyone wants morning snacks, I have them here. Do not take more than 2 handfuls of this, otherwise you'll overdose on some. Still missing a couple people. Oh, thanks. I know Hayun was in a meeting before this, so I guess these are light.
I'm still waiting for a couple dissertation committee members. So.
Do the committee members just want to check the front seats?
I guess the last 2 will have to.
Yeah, it's here. I never liked the layout of this room, like when taking classes, because these 2 seats just can't see this at all. Yeah. Let's see. Any emails? No email from. Should I just start in Michigan time regardless? Nothing like front row seats. Have you seen Laura on the way down?
Sorry?
Did you see Laura on the way down?
I did.
Sorry.
I will be lonely the first row.
Yeah, it's not gonna work. I still have to wait for Laura, I guess.
Somebody tried calling me? I didn't get my phone, unfortunately. Do you have it, Lily? Is that the good one? Okay.
With the 277 number.
Sorry?
Is there text with the 277 number?
Yeah, that's not office and that's not home, so it's just feel like a hazy man.
That's hot water. Where are we?
That's good.
Sorry about that. Oh, my middle.
Okay, we can start.
Okay, we can go ahead and get started. So thank you all for coming for Chris' dissertation defense. For those of you who don't know me, I'm Maureen Sartor, a co-mentor of Chris's along with Jean Zhou. So Chris came to us from the University of Maryland, where he got his undergraduate degree in mathematics, or at least that's what I thought his major was until recently. We found out he actually had a double major in math and cell biology and genetics. So since he's been here, he's mainly been working with Jean Zhou and myself after receiving his master's in biostatistics in 2015. And so he's been very productive while he's been here. And I thank him for helping out with other projects in my lab that we won't be talking about today, including also some collaborations with biologists, and he has a published paper from that work as well. He's also gone to and presented at several conferences since he's been here. So for me, the most memorable was when we went to Prague to the large annual bioinformatics conference, Joint ICE and BNE-CCB. But if I had to guess what his most memorable one was, I might guess the one that had nothing to do with his thesis work, the statistics in esports. And so also along the way, he received the Mijar statistics grant. And that was the only grant he ever applied for, so he's got a 100% success rate.
Time to retire.
It's way higher than my success rate. And so with that, I'll hand it off to you guys.
All right, thanks everybody for coming. Today my thesis will be improvements and developments in gene regulation and single-cell gene expression data analysis. So before we begin, a lot of people think of genetics simply like this. You have a gene, and then it becomes a protein which then does some function. Without this gene, you don't have a function. However, this is not the entire story of how genetics works. As DNA first has to be transcribed to mRNA, between the steps of DNA to protein to function. So even though if you have the gene, if something happens in these intermediate steps, you also don't get a protein or function. Alternatively, there are also times when we can see much more mRNA, which sometimes results in more function. So overall, my talk will be about regulatory genetics and transcriptomics. So compared to regular genetics, instead of asking if we have the gene or no gene, we assume we have the gene, and we ask how the genes are regulated or turned on and off, and how much it is expressed. Here's the outline of my 3 projects under the little theme that I just introduced. All right, let's get started with the first one. We want to know how genes are turned on and off, and one of the mechanisms that do that are transcription factors. So transcription factors are proteins that just bind on the genome, often near a transcription start site, that increases or decreases the activity of RNA polymerase, which does the transcribing. And transcription factors can bind all over the genome. And so we need something to be able to measure the activity of these transcription factors. To do that, we have ChIP-seq. So what ChIP-seq does is you pick a particular transcription factor, and ChIP-seq will output a bunch of peaks all across the genome. And ChIP-seq will allow us to answer things such as where the peaks are, how many peaks, how strong the peaks are binding, and usually how strong the peaks are binding are designated by pretty much the area underneath the peak, as you can see from the real data on the bottom corner. With several peaks across the entire genome, we still need to link them to target genes. So when TFs are binding really close to a gene transcription start site, we can pretty much assume that that gene is its target with pretty much no questions. However, most of the time it's not so obvious. If a transcription factor binds somewhere in the middle between 2 genes, or just anywhere not close to a transcription start site in general, where do we want to assign this transcription factor? So in order to handle those, we need to create things called gene locus definitions. So first we define the gene locus as the region on the genome where if a peak lands into this region, that peak will be assigned to the gene. And the locus length would just be the length in base pairs of that locus. And then the entire locus definition would be just the combination of every single gene's loci. So here we have some examples in the bottom. One of the ways we can define a locus definition is just to take 5 kilobases around the nearest transcription, around any gene's transcription start site. So we only care about the peaks that are binding close to the gene transcription start sites. So if you take a look at peak number 2 under this locus definition would not be assigned to any gene. Now, alternatively, we can also define a locus definition such that any peak on the genome is then assigned to the gene which has the closest TSS. And this nearest TSS locus definition is one of the ways we can assign all of those peaks that are not away from, or that are really far away from gene transcription start sites. Now, talking about genes one by one is great, but however, usually biologists like to talk about sets of genes in the context of a pathway. So if you take any biological pathway, for example, we can take cell cycle, there are a bunch of genes that are relevant to cell cycle, and you just take all of those genes and make it into a set. With a gene set, we can perform something called gene set enrichment analysis. That basically tests if genes in a gene set has more or less of a signal than genes not in the gene set. So if you take some example, if you take a gene set whose useful size is usually between 10 and 2,000 genes, and suppose 10% of those genes has a mutation. And if we take a look at all the other genes, and we see that only 0.1% has a mutation, of course we can suspect that 10% is probably larger than 0.1%, and we can test for this. And if it is significantly larger, then we can say that this gene set is enriched for mutations. So now that we have gene set enrichment and ChIP-seq data, here are some existing methods for ChIP-seq data to measure peaks in transcription factor activity. First, we have ChIP-Enrich, which tests how many peaks there are, but it's in a binary sense. So it only looks if a gene has at least one peak or more. And so we would be modeling the probability that the gene has at least one peak. We also have broad-enrich, which tests how wide the peaks are. Usually these are not for transcription factors, but they are more for histone modifications because they tend to bind in a wider area around the genome. And finally, we have GREAT. And GREAT counts how many peaks there are, but the ChIP-Enrich manuscripts show that there's some high type 1 error rates. So we want to improve on the how many peaks test. So my first project, I developed Poly-Enrich, which, similar to GREAT, also counts how many peaks there are in a gene set. However, we made some modifications to correct the error rates. And as of now, the Poly-Enrich manuscript is still in a revision at NAR Genomics and Bioinformatics. So both GREAT and Poly-Enrich both count peaks. So what's the difference between them? So GREAT only assigns peaks to the gene sets without accounting for which genes they're assigned to. So it's missing like an intermediate step. So Poly-Enrich, we take a look at the peaks on the genes, and then the genes are assigned to the gene set. By doing this, we can use the information about the genes, like any information about the genes, and adjust for any gene heterogeneity. An additional motivation since we already have a binary test, why would we want a count test? So I will give you some scenarios to show you why a binary test may not be the best at all times. So here we have this first scenario, this cartoon scenario where we have 3 genes in the gene set and 3 genes not in the gene set. And we see that 2/3 of the genes in the gene set is assigned at least 1 peak and only 1/3 for not in the gene set. So ChIP-Enrich, the binary method, would be comparing 2/3 versus 1/3. Meanwhile, Poly-Enrich, the count-based method, would be comparing 3 versus 1. In this scenario, both methods are doing reasonably well. Now, in the second scenario, we add a couple of peaks to the genes in the gene set. However, they're the same genes as before, so ChIP-Enrich still only looks at 2/3 versus 1/3. However, more peaks might mean something, and using a count-based method, we'd be able to compare 5 versus 1 instead, which is possibly a better fit. And finally, suppose we have this case where every single gene is assigned at least 1 peak. So a binary method would not be able to detect any significance at all in this case. And so we have to use the count-based method for this scenario. So now I'll compare the models between the binary and count-based models. So as a binary model, ChIP-Enrich models the probability that any gene has at least 1 peak. And in contrast, Poly-Enrich is a count-based model, so it models the average number of peaks. So the main difference between these two models is that, as modeling a probability, we want to use a logit link, and modeling the mean, we want to use a log link. And in both models, we have this gene set indicator here, which is 1 if gene I is in the gene set, and 0 otherwise. And beta 1 is our parameter of interest. And so we would interpret this as, in ChIP-Enrich, if beta 1 is positive, then if a gene is in the gene set, then there is a higher probability that this gene is assigned at least 1 peak. And in Poly-Enrich's case, if beta 1 is positive, then if a gene is in the gene set, the average counts of the gene is likely to be higher. And finally, we have these F and G terms, which are spline terms to adjust for locus length, which was shown to be a confounder in both models. So if you ever work with splines, you would remember that they aren't really the fastest at being computed. So we noticed that since the gene set is usually less than 10% of the total number of genes, having this gene set term doesn't really affect the entire fit as much. So if we compare the spline generated without the gene set term and with the gene set term, you can barely tell that there's a difference in the spline. So since we have to run this model for every gene set, which can be up to thousands, and every single one we have to do a spline calculation. But if they're almost all the same every single time, why would we bother recalculating them all the time? So instead, in order to save time, we estimate the spline once, and then use that spline estimate in every single subsequent calculation afterwards. So to do this, we first model without the gene set term, and then using this spline term, we can find the predicted values for every gene, and then we just use this prediction as a linear effect in the model, which would result in, for 4,000 gene sets, instead of calculating 4,000 splines, we only calculate 1, which saves 70% of runtime, which is pretty significant. So now we have two ways to count peaks, but how many? But in reality, how many peaks how many binding sites do we really need to regulate a gene? So we can ask, is one peak sufficient for maximum regulation, and multiple peaks on top of this won't add any more regulation? In this particular scenario, the binary method would be a better fit. Alternatively, we can think that maybe more peaks results in incremental regulation. Like two peaks is more regulation than 1, 3 is more than 2. So between our binary and incremental methods, how do we know which one is optimal? If you know the mechanisms of a particular gene set, you could make some educated guesses, but most of the time we really don't know. We found that even within a single ChIP-seq experiment, there are some gene sets. If you take a look at the plot on the right, which is a plot of several gene sets under this experiment, we see that several gene sets perform better with the incremental model, and some gene sets perform better with the binary model. So because of this, the takeaway is that there's really no one-size-fits-all model, since transcription factors are just too varied, and there's just really no way to predict this. So one of the workarounds we did, which I won't cover in the essence of time today, is that we can combine multiple tests to cover more options. Well, the summary for project 1 is that we developed an improved gene set enrichment model for counts. I decreased the runtime of both ChIP-Enrich and Poly-Enrich with the spline estimation, and the resulting summary is that there's no one-size-fits-all model. Any questions for this first project before I move on? Yes, Mike.
Chris, are there any meaningful differences in results between the one spline fit and fitting splines for all the different gene sets? Because saving 70% of the time is nice, but it's not a big deal from my perspective if the fits you get are quite different. But if they're quite similar, then
they're pretty much exactly the same. So if you take a look at like beta 2 is almost always like close to 1.
Okay,
Yeah.
thank you.
There's two questions from your viewers. One, does model specification affect the results or conclusions? And two, is there a rough idea or explanation of why the spline portion is the same for multiple gene sets?
Okay, I'll answer 2 first. The question was, is there a rough idea or explanation why the spline proportion is the same for multiple gene sets? And the answer to that is that usually the gene set term is 1 or 0, and it's 0 for 90% of the 90% of the time, which is why it matters. It doesn't matter as much. The first question: does model specification affect the results/conclusions? Why, yes, because depending on the gene set, for biological reasons, some genes are just regulated within a binary sense, and some genes are just regulated in a count sense. So if you just go into analysis blind, you really don't know which method you're supposed to use. All right, any additional questions?
Do you ever expect the observed range of these counts, or how the counts are distributed? So
You mean like a
yeah, it's interesting to see the binary and the additive models are working different in different gene sets, and I'm just curious that if there's, for example, if there's some transcript factor that happens a lot of time, maybe so you might think about some hybrid model with 1 and 2 matters, but 1 and 2, 3 matters, but all of that doesn't matter, or something like that. So you are currently linearly modeling it. So having 1 and having 10 is 10 times more
Yeah.
Score.
Oh, so you're thinking if maybe there's like another way that instead of counts going up linearly, there's some other like function.
Right. I mean that's not that's a start of my question. But my question was simple. So how many cases, how many gene transcript factors or gene, how many of these units have more than one more than one count, so that the binary and the non-binary.
Oh, so usually when the if you remember the third scenario of here,
Right.
like are you talking about like how often this happens?
Right.
So usually this happens when there are hundreds of thousands of peaks, or this is more likely to happen when there are hundreds of thousands of peaks. And there are some experiments, not transcription factors, but for other things such as repetitive elements that have millions of peaks. And so when there are millions of peaks, almost all of the genes are assigned at least one peak at that point.
So is it how often, how frequent is it?
So it depends on the data you use. For transcription factors, it's generally not as common. I think only in the data we used, only around 1 or 2 were large enough such that this was the issue. But in repetitive elements, it's pretty much almost all the time. Any other?
Yeah, so does multiple peaks mean that the same transcription factor is binding in multiple sites near this gene?
Yeah, so same transcription factor is binding in multiple sites in this gene.
So they don't tend to all bind in the same place, because that would just result in greater peak intensity, right?
Yeah, they bind like close to the same place, but not
Okay.
exactly the same place.
And you don't merge those together?
Depends on, so I guess I'll go back to here. Depends on how you define a peak in your peak calling. So you practically pick like a horizontal line as your like threshold for your peak. And so if you pick a high threshold, then like one peak can become 2, or if you pick a low one, then multiple peaks can become 1. Now we can get started on project 2. So transcription factors often bind near a transcription start site. We call this region the promoter region. And like when I said, if a transcription factor is binding in a promoter region, we can pretty confidently say that that is its target gene. Now, of course, transcription factors can also bind really far away from gene transcription start sites in scenarios such as this, when the DNA bends over itself and the transcription factor helps RNA polymerase bind there. So we have these two types of binding, distal and proximal binding, where proximal is close and distal is far. And in this cartoon case, we would see that there are 3 peaks that bind really far away from the gene. And these regions, these faraway regions where peaks can bind and still regulate genes, we call them distal regulatory elements, or DREs for short. If the peaks in the DREs upregulate, we call them enhancers, or if the region upregulates, we call them enhancers and silencers otherwise. Now, most people call them enhancers, but for this talk, I'll be calling them DREs. Now, most gene set enrichment tests do not test where the peaks are within their own gene loci. So if you take a look at these 2 scenarios and consider Poly-Enrich, in Poly-Enrich's viewpoint, these 2 scenarios would result in seeing 3 peaks for this gene. However, we can obviously see that these 3, in the first scenario and the second scenario, are very different in where they bind. So we want to create a method to distinguish between these 2 scenarios. And so we can answer, are the TFs regulating the gene set through the promoters or through their enhancers, or DREs? So I developed this method, proxReg, which stands for proximity to regulatory regions. whose main goal is to test how close peaks are to regulatory regions. And the manuscript for this one is also in revision for Frontier Genetics. So overall, proxReg is a reasonably simple model. For any gene set, we take a look at all the peaks in the genes, and we basically test if they're significantly closer or farther to a regulatory region. So if you take this scenario, this histogram here, the red are the peaks in the gene set, and we can see that there is a reasonably obvious shift towards 0, which means the peaks in this gene set are closer to the TS, or are likely closer to the TSS. So to test for this, we use a Wilcoxon rank sum test on the peak distances, but we first need to adjust for the gene locus length confounding, similar to how we did for ChIP-Enrich and Poly-Enrich. So overall, the model works like this. We first take every single peak and calculate their distances to the regulatory region of your choice, either a TSS or a DRE, one at a time. We then model a spline with the confounding factor in the log scale. And then we calculate a new adjusted distance. That is the old distance minus the distance to the mean from this fitted spline estimates. And then we can use the Wilcoxon rank sum test on these adjusted distances. And of course, since this is done for multiple gene sets, we then adjust for multiple testing. So to make sure our method is working, we chose a pretty known transcription factor 6.5, which has already been proven by other biologists to bind near transcription start sites. So we ran both Poly-Enrich and proxReg on a 6.5 experiment. And the results will look something like this. For any particular gene set, we'll get a Poly-Enrich status and p-value, and same for proxReg. And from this positive correlation, we notice that if Poly-Enrich detects more peaks, then these peaks are also more likely to bind closer to TSSs, which confirms this finding that we've had before. Now this was just the easy case, and we have some more fun cases. Some transcription factors are able to bind in several different ways. So PU1 is an example where there are gene sets where they bind, where PU1 binds closer to the DREs, and some gene sets where they bind closer to their TSSs. So this is some really useful information that we can use to complement a pre-existing gene set enrichment test to enhance our understanding. So I'll use Poly-Enrich as the example of the gene set enrichment test. So suppose we have a bunch of significant gene sets from Poly-Enrich, and then we also look at their proxReg results. So suppose proxReg says that this gene set, the transcription factor tends to bind closer to the TSSs. Then we can pretty reasonably say that this transcription factor is regulating this gene set directly through their promoters. And on the opposite side, if proxReg says that they bind significantly closer to DREs, then we can say that they're regulating through distal binding. However, if proxReg is not significant, then there are multiple of other explanations. One of them could be this transcription factor just happens to be regulating through both distal and proximal binding, or it could actually just be a false positive, either from the peaks themselves or from the GSE test. So the takeaway for this project is that we want to add different perspectives to increase our insight. So many methods really only look at one perspective. Like Poly-Enrich only looks at the number of, or only tests for the number of counts, and proxReg only tests for the distance to regulatory regions. However, if we combine the results, we can take a look at a more complete story. So the overall summary is we developed a method to test for proximal binding. And if we use this as a complementary test to other gene set enrichment tests, it allows for the better understanding of the transcription factors behavior. One current downside that we've had is that for distal binding, usually there are other genes in between the distal binding TFs and its target gene. So in this case, if we were to naively assign all the peaks to their nearest gene transcription start site, these three purple TFs would be misassigned to the first left gene over there, the first green gene, left green gene, down at the bottom, and not to the actual target that we want to. So another project that I worked on, but I will not go into much today, is that we created several additional locus definitions to be able to assign distal peaks to their correct target gene. And to do this, we basically took several sources of DRE locations and several sources of DRE targets, and then we used them to create several locations on the genome and assigned these locations to some target gene. And the overall conclusion for this project is that we managed to create 10 or so locus definitions, 10 or so good locus definitions that improve the gene set enrichment results, compared to just naively assigning to the closest gene TSS. Open for questions for part 2. Yeah.
So in ChIP-Enrich, you have, and in your I assume with your new method, the poly-enrich, you have subsets of the tests which can only look at the peaks within less than 5 kb away from TSSs, right, which would basically cover peaks within promoters. And I don't remember if you also have tests that are greater than 5
Yes, yeah,
kb
that exists too.
away. So those crudely divide up the peaks into peaks in promoters and peaks not in promoters. Do you see similar results of those two, when you compare the p-values from those two tests, do you come up with similar conclusions to when you use proxReg, or what is proxReg capturing that those two tests might not be?
Okay. I personally haven't tried comparing between promoter and not promoter regions. At least for transcription factors that are well known to bind close to TSS, I would assume that the results would look pretty different, as when you remove all of the promoter bindings, you probably remove almost all of the peaks themselves. However, of course, in the more interesting cases, if you only take a look at, so I guess if there is a gene set that does bind to both promoters and DRE regions, then I can imagine that these two results would be similar between two locus definitions. They both might not be as significant as they should, but I can see them being of similar significance because of that. But in terms of comparing with the proxReg results, proxReg would also say it would be insignificant. So I suppose the better question would be if we compare poly-enrich only with promoters with proxReg, and then compare poly-enrich only with not promoters with proxReg. So I think doing so would be able to solve the issue of, so if we take a look at the middle row here, so if we suppose this is actually what's happening, that some TF is regulating through both proximal and distal binding, then yeah, I suppose if we rerun Poly-Enrich except only use promoters and then rerun Poly-Enrich only with enhancers, we'd be able to distinguish between if this is actually the true case or not. Does that answer?
It does. It's just you've got two different tasks that I think are getting at similar things, and I was just curious whether you could make an argument that no, proxReg, there's right, it's different in how it does that correction. It's a little complicated, right? The correction is different.
So I think for promoters, if you only test for promoters, I can see the argument that it works fine. However, when we look at enhancers, when we look at DREs, the DRE locations are still scattered across the genome. So even if we use Poly-Enrich, they still might be far away from the DREs, and we don't know. However, in proxReg, we actually look at where all the DRE locations are, and we can actually measure. So if the peaks are actually really close to the DRE locations, then it would be able to get more information than Poly-Enrich would.
Yeah, absolutely.
Okay. All right, now we can get started on project 3, which talks about transcriptomics, which helps answer how much a gene is expressed. So we want to be able to measure how much a gene is expressed. One of the ways we can do that is just simply through RNA-seq. RNA-seq just literally counts how many RNA transcripts there are in a scenario. And that is one of the ways we can somewhat use to measure gene activity. And we can use this test called differential expression, which tests how many more or less transcripts there are. So in this case, small example, we have 4 cases and 4 controls. And suppose we have this data, this RNA-seq count data. And we can see that, the cases seem to have a higher number of counts in the cases. So if the case samples are significantly higher than the control samples, then we can say that this gene is differentially expressed in whatever scenario the case is. Now recently, there is a new technology called single-cell RNA-seq. So traditionally on the left, you would run RNA-seq on a bulk tissue which contains thousands of cells, but for illustration purposes, there's only 4 here. So it would take all the cells and just add up all the transcripts within all the cells. And so bulk RNA-seq would say that there are 9 transcripts in this tissue. In single-cell RNA-seq, they would separate every cell individually and count out the number of transcripts individually for each cell. So the single-cell RNA-seq would be able to split up all the counts for all the cells. Now, why are people so excited about this technology? So because we can separate into different cells, we can also categorize the cells into different cell types. And doing so, we can ask questions that we couldn't before with bulk RNA-seq. So some examples are like, is there a difference between 2 cell types? I guess example, do neuron cells have a high expression of some gene than skin cells? And we can also stratify within cell types. So within a particular cell type, like within neuron cells, do Alzheimer's patients have a higher expression of some gene versus non-patients? Now, if you look at this last case here, you would probably have a design, something like this, a hierarchical design, where you have several individuals in your cases and several individuals in your controls, where every individual themselves has a large number of cells. Now, you can be naive about this and just ignore everything about the individuals and only pretend cells are just independent cells. So if you were a naive statistician, you would basically compare 15 case cells and 17 control cells. However, this is generally a pretty bad idea. So one thing that happened to me was that there are sometimes some individual just happens to have a lot more cells than everybody else. And not only that, this individual also, for some reason, has a lot more counts as well. So ideally, what the actual effect sizes are is you would take the means of the means for the individuals, and then you would compare them. So the cases, we would have a mean of 9, and the control, we have a mean of 11. However, if we ignore everything about the individuals, this one particular individual, which has several cells and several high counts, will basically dominate the entire analysis. And so if we take the mean of only the cells, ignoring the individuals, we actually get that the mean of the cases has jumped up to 11.7, which is now larger than 11. So ideally, we would want to use a mixed effects model like the one used in PQLseq. So the model is generally a Poisson mixed model, given counts for given your n cells and you have p individuals. We have a mixed effects where your fixed effects are here for your covariates of interest. Your case control variable would be here as well, and other adjustments of interest. We have an individual random effect and an individual and a cell random effect. So mixed effects models are generally doing pretty well. However, they do not scale very well to larger data. And since single cell RNA-seq is on the order of tens of thousands of cells, if we ran PQLseq on some single cell data, it would take on the order of like weeks and months, which is practically not practical. So thankfully, I figured out a way to use a low-rank property of a matrix to decrease the time complexity of this algorithm. So in PQLseq's estimating algorithm, there is this matrix H that needs to be inverted, which, if you know, is of order of n cubed, which is pretty slow. However, and we see that since we see that D is diagonal and the identity matrix is diagonal, we have a diagonal matrix plus another matrix in the middle here. And this matrix ZZ transpose is special because if we define Z as an n by p matrix, such that Zij is 1 if cell I belongs to individual j and 0 otherwise, then ZZ transpose is an n by n matrix, but it only has rank p, the number of individuals, which is a lot smaller than the total number of cells. As we have now we have a low rank matrix, we can use something called Woodbury's matrix identity, which allows us to invert a matrix, as so. So we have H inverse is equal to this big calculation. Now we see that there is still an inverse here. However, this matrix is now only a p by p matrix. So big O of p cubed is, since p is a lot smaller than n, is pretty much negligible compared to the rest of the algorithm now. So to show that this works, I simulated a bunch of data of increasing sizes, and we do see that I simulated a bunch of data sets for 10 genes of increasing sizes, and as expected, we see as we increase the number of simulated cells, the time increases. However, we see that the lower rank version increases much slower, or increases much slower than the original version. And since usual data sets are even beyond this table, you can imagine how long it would take for the original version. And so this is great because most investigators do not have hundreds of thousands of computing power. So they can now use this algorithm as a more powerful method without sacrificing pretty much a month of their computer. So now that we showed that we can actually use this, we want to see how this algorithm performs on data. So I created some type 1 error and power simulations, and I based these simulations on the mixed model as PQLseq is defined on. And I simulated 5 effect sizes, 5 increasing effect sizes. and I use beta equals 0 as the empirical null that we would use for type 1 error. Of course, we want to compare to other differential expression methods, so here's a list of the ones I compared with. Now our control model is basically plain linear regression, and the thing I want to mention here is I use a nested design to account for the hierarchical structure. Now for any other method in this list that can adjust for several covariates, I also use this same nested design in the model. That way we can at least we can have a more fair comparison between like the mixed model and a fixed effects model, because otherwise it would actually just be kind of unfair to just use the other models with poor assumptions. So the first thing we saw that here's the type 1 error plot on the null distribution. We see that our method is the most conservative of them all. However, some of the methods are inflated, as we see here, and some other methods are also conservative. Now, in the original manuscript for PQLseq, that they also discovered that it is conservative. However, it is an artifact of a small sample size, which is also similar to the sample size that I use to simulate this. And we'll get back to the this type 1 error later. So to test for power, I don't want to just use at unadjusted p-values, because if you pretend some method is just highly inflated, and then you run it on some simulation where you expect some significance, then since it's already inflated, everything will be significant, and then this method would have like perfect power, which would be not a very fair comparison. So to do that, I created a 5% empirical threshold for significance. So if you take all the null p-values and order them and take the 5th percentile of them, that p-value would now be the threshold for significance for that method. And so that way, even if a method is inflated, it would require a lower p-value to catch significance for the power simulations. So how did our method do with the power of? We see that our method has the highest power amongst all other methods. The only thing of note is that edgeR aggregated does the worst, because I use this as a second control where I do not use the assumptions, or I do not use the individual assumptions. I instead imitate what bulk RNA-seq would do by summing up every single counts in every single cell in every individual, and then just running edgeR. So simulations are nice, but of course it's unfair to the other methods, because if you simulate based on your own model, you hopefully should get your own model to be the best. So I have some real data by Justin Colicino, where we have eight African Americans and 7 European Americans, and we have almost 7,000 of a single cell that they pre-filtered, and we want to test for differential expression on race. And to get a null distribution for the real data, I just permuted the individual labels for the cells. So every cell would be assigned to a different person instead. And of course, with the person would also be assigned the race label. So in this simulation, in this permutation for real data, we see that if you can see, you can see the black line for us is doing really well, while everything else, especially these four, seem to have a super inflated p-values. Now we use the same method to calculate power, and so we have that our method does show the most power compared to every other method in real data. Of course, you see that since this is times 10 to the negative, not shown here. These are times 10 to the negative 3. So the general sum pretty low power. However, I believe the low power is just due to the data probably not having as many really differentially expressed genes, and there's a kind of a small effective sample size too. But the point here is that our method is still the most powerful compared to all other methods. The summary here is that, of course, using a random effects model would increase power for the hierarchical design, because that's what you should do. However, we also made it so that investigators who do not have access to a lot of computing power can properly use their assumptions to create use a more powerful model. And finally, the current time complexity of this algorithm is n squared from matrix multiplication, and there might be some ways to still improve that. As this method is still slower than most of the other methods. However, it is no longer extremely slow. Thanks for any questions for 11 o'clock?
Did you check the gene whether those differential expressed gene make sense?
Yeah, for the real data, right?
Right.
Yeah. They make sense.
And you calculate the significance using permitted as a null distribution, so that's why.
Yeah. So that's why some of the methods, especially with the inflated ones, have like pretty much close to 0 power. Okay. What is that? Yeah, okay.
Sorry. Oh, maybe not the last answer.
Yeah, but I'm done.
Oh, okay, sorry. So question for you, how does PQLseq account for the over-dispersion and non-inflation characteristics of single-cell?
Okay, so yeah, so the question is how does PQLseq account for the over-dispersion and zero inflation characteristics of single cell? So one thing, let's scroll back up to monorator. So one thing of note in single cell is that because single cell technologies are not so perfect yet, there tend to be a lot of dropouts, which results in a lot of extra zeros. This version PQLseq does not quite account for the zeros as well. So it counts for the variant, the over-dispersion, using these random effects. The random effects are used to account for over-dispersion. So if there is more variance, then this sigma squared term would be increased. However, in terms of the zero inflation, some people argue there is a post somewhere that argues that you do not necessarily need to use zero inflation in your single cell models at all. So this goes back to the theme over in the first project, where you have like multiple methods, and some methods of course would work better on other methods, and so there are single cell data sets that do have a lot more zeros that would do better if you had a zero inflated model. Yeah, that would do better in a zero inflated model, and there are some single cell data sets, hopefully in the future, when dropouts aren't really much of a concern anymore, where we no longer need to account for zero inflation. Any other questions?
Let's say there is a mixture of multiple cell types.
Mm-hmm.
Okay. And if differential expression in bulk data set, if there's a difference in the cell type composition, actually that could create a false differential expression.
Oh no, sorry.
So let's say there's there are two individuals. One individual has a cell type A enriched, the other individual has a cell type B enriched. So if you mix them together, so if there's a difference in the cell type proportion, that might create a false enrichment of differential expressed genes.
Oh, sorry, you're saying if two individuals have different proportions.
So that's a known problem for the bulk RNA-seq, and the single cell RNA-seq is supposed to be able to correct for it. But I wonder whether you or method would be able to correct for it.
Yeah, it should be able to adjust for individuals. So if we go, I'll use this this page here. So you suppose this individual has more of this type of cell, and this individual has more of another type of cell.
But you're not testing you're testing across all cells, right?
I mean, it depends on your design. You can stratify by cell type, or you can not, or you can add cell type as an additional.
But your test does not explicitly account for the cell types. So
it can. You can add an indicator to adjust for that. Yeah. Looks like we're good. All right, so now we can start on the final conclusions. So the overall theme of my dissertation was that it's important to not only look at if genes are present, but how they are regulated and how much is expressed. From the first project, we see that some methods are better fits than others, but we are generally unlikely to know which one is optimal. From the second project, we found out that we want to look at data from other perspectives to provide a more complete story. And from the third project, we see that there's a balance between how complex your model is going to be and and how long it's going to take. Now again, to some future work and ideas that I started but didn't complete for anybody who wants to take these on. So on the theme of looking at different perspectives, we had this idea where, since transcription factors can bind to genes for reasons unrelated to the context of your experiment, and genes can be expressed, but they're actually like downstream signals from the actual gene that is causing from like the first gene that is causing the function. So if we combine analysis from ChIP-seq and RNA-seq, we can use this table to sort of find the truth between if a transcription factor is truly relevant to the context of your choosing. And secondly, there's another project where suppose you have a gene-by-gene correlation matrix, and then you group them like this, such that all the genes, you pick a gene set and group all the genes in the gene set to the top. Now for a gene set that we want to enrich, we want to be significant, all the correlation values in A should be close to 1. All the correlation values in A should be close to 1. All the values in B should be close to 0, and C should be generally random. And so we want to find a statistical test that's powerful but not slow, because I have powerful and slow, or fast but not powerful. So we want to find another method that balances these two again to be able to answer for this. And so these two are the extra unfinished ideas, if anybody else wants to tackle them. And here, any final questions?
Yeah, one last question. Question from topic number 3. How important is the use of random effects?
Okay, I get it. So the question is, back in project 3, how important is the use of random effects? Because you could just model the cell and individual cells as a fixed effect, and that should make computation faster. Okay, so the question is, how important is using a mixed effect? And I'll go over here. So the way you can use a fixed effects model using a nested design to imitate random effects model. And of course, this does run faster than using a mixed effects model, at least with current algorithms to estimate mixed effects. And the question is, just model the. So how important is the use of random effects? So I think the only difference would be that without the individual random effect, you might have individual fixed effects diverging to large variances, and if you had a random effect, you'd be able to minimize the individual effects.
Try models?
Yeah. So every model. So like so linear regression, EdgeR, Lemma, and DCEQ2. I used like the linear workaround to compare and. I used the linear workaround to compare, and I got that. The random effects model does indeed perform better.
So is PQLseq the only random effect model?
There are. I saw some other random effects models, but they were not for single cell. Trying to think of the one I see, which one it was, because I remember I saw a few, but they were not able to be used on.
What are the ones you compared?
Oh yeah, no. All of these are all fixed effects models. Any other questions? All right. Thanks everyone for sticking with me. Okay.
So do you have this room reserved until?
12. I think I have this room until. The next thing is 1:30, so we can go over time.
Chris.
Okay.
Congratulations.
Nice.